Distance between the two strands is equal to 20 angstrom. Okay, so with these information, let us draw the structure of the DNA. Let us start with the sugar. Phosphate is attached here. This will get attached to the next sugar. So this is the carbon of the next sugar. Oxygen, carbon, 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 and likewise goes like this. Let's number this. Carbon 1, 2, 3, 4, 5. Carbon 1, 2, 3, 4, 5. So you see on this end, all the sugars, they have their fifth carbon on this end. So this is the 5 end, and all sugars have their third carbons on this end. Okay, so the 5 end to 3 end direction is this in this particular strand. Okay, now let us put nitrogen bases to these sugars. So I'm drawing a nitrogen base here, and then you need to tell me what nitrogen base I'm drawing here. Carbon, carbon, CN, CN, C, N, C, N. Okay, and on this carbon here you have NH2. This carbon has a hydrogen. So let's balance this. 1, 2, 3, 4. 1, 2, 3. There's a double bond here. Then we have nitrogen, so there's a double bond here as well. So this carbon also has 1, 2, 3, 4. Right, so this is which nitrogen base? This is adenine, right? Okay, let us put another nitrogen base to this sugar here. Let us put guanine. Okay, so likewise C, C, N, C, N, C, N, C, N. In guanine, this carbon has a double bond oxygen here, and this carbon has a amino group. So NH2, let's balance this. 1 2 3 4, 1 2 3, nitrogen, carbon is saturated. This carbon is saturated, therefore we cannot have a double bond on this nitrogen, so we have a hydrogen here. Okay, and this side, so we have a double bond here, double bond, and there's a hydrogen here. Okay, so similarly there's a double bond here as well, and there's a hydrogen here. Right, so this is one strand ready, and we have the sugar phosphate, sugar phosphate. So this is sugar phosphate, sugar, alternating sugar and phosphate. So this goes up like this, and this nitrogen base here, what we've drawn here, this is guanine. Okay, very nice. Now let us, this continues. Okay, so now let us do the other strand. Similarly, we'll do the other strand. Let's start with the sugar again. So here we have the sugar. Now what have we seen? We have seen that the two strands are anti-parallel, right? So oxygen, carbon, 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 and then there's a carbon here, phosphate group, this attaches to a carbon, and this is which carbon? This is the third carbon. Carbon, 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 the fifth carbon, and so on. Right, here again, this is the phosphate. So what we have here? Here again we have a sugar, phosphate, sugar, alternating units, and what is the direction here? So let's label. So this is carbon 1 2 3 4 5, 1 2 3 4 5. So every time you see the 5 end, the fifth carbon, is on this side. So this is the 5 end, and the third carbon is on this side. So this is the 3 end. So what is the direction in this strand? It is the reverse. That is why this is anti-parallel. Now let's do the nitrogen base on these sugars as well. So what have we seen? We have seen that adenine binds with thymine. So let's do a thymine here on this sugar. It binds on carbon 1, carbon, carbon, N, C, N, C. What is the thymine structure having? It is having an oxygen here. It is having an oxygen here, and this one, there's a methyl group here, There's a hydrogen here. Okay, so this is this thymine. What else does this require? This nitrogen has 1 2 3, this carbon has 1 2 3 4, carbon has 1 2 3 4, therefore there has to be a hydrogen here. So this is our thymine. Okay, next let us see. So we have guanine here, so this will bind with cytosine. Let's do a cytosine on this sugar. Cytosine is again a pyrimidine, so we have two carbons, N, C, N, C, and cytosine. There's an amino group on this, NH, H, okay, and there's a double bond oxygen here. You have a H, you have a H here, and then here. So is everything balanced? 1 2 3, 1 2 3 4. This one would be a double bond here, right? So this is our cytosine. Cytosine. Perfect. Now let us do the hydrogen bonds between the nitrogen base pairs of the two strands. Okay, so this is what, there's a phosphate group here. Okay, okay, then this goes. So right now let us do the hydrogen bonds. So between adenine and thymine we have got two hydrogen bonds. Now what is a hydrogen bond? A hydrogen bond is when a hydrogen that is attached to a electronegative atom. So let's say here this hydrogen which is attached to this nitrogen, this will experience a pull from this highly electronegative atom, that is oxygen. Okay, and then the bond that will be formed is this. So this is one hydrogen bond. Likewise, this hydrogen attached to this nitrogen experiences a pull from this nitrogen, and thus it is shared between these two nitrogens. So these are the two hydrogen bonds formed between adenine and thymine. So adenine and thymine, they are binded or bound with two hydrogen bonds. Likewise, guanine and cytosine. So here this oxygen, it exerts a pull on this hydrogen. So there's one hydrogen bond, and this hydrogen experiences a pull from this nitrogen, so it is shared there. Another hydrogen bond, again this hydrogen experiences a pull from this oxygen. So this is three hydrogen bonds. So there is guanine and cytosine, we have got three hydrogen bonds. Okay, now in case you are wondering why this hydrogen is not experiencing a pull from here, so hydrogen bonds has to have two highly electronegative atoms which are pulling on hydrogen from either side. So here this hydrogen is attached to the carbon here, which is not a highly electronegative atom. That's why there won't be a hydrogen bond being formed in this case. Right, so now what we see? We see these two strands here, and we can actually, we can extend this. These are the two strands. Okay, before we proceed with regards to the rest of the strands, let me just ask you, what are the bonds that you see here? So the bonds that you see here between this sugar and this nitrogen base pair, what is this bond? This is N-glycosidic bond. Okay, N-glycosidic bond. You remember when we were doing carbohydrates, we had seen O-glycosidic bond. So here this is N-glycosidic bond. Okay, we have not put the hydrogens and the hydroxyls on the various carbons of the sugar. So but I'm sure you know that on the first carbon there is a hydrogen, there is a OH, and on this nitrogen of this nitrogen base there is a hydrogen as well. So H and OH, they water moves out, and then there's a N-glycosidic bond that is being formed here. Okay, this is one bond. Okay, next, between the phosphate and the sugar, what is the bond that you see here? This is the phosphoester bond. So that is between the phosphate and one sugar. Now this phosphate, you see, this is joining these two sugars. So this sugar and this sugar is being joined by this phosphate here. So what will be the bond that will be formed? In this case, it will be the phosphodiester bond. Okay, so when this phosphate, it joins this sugar and this sugar, this is because there are two esters, phosphodiester bond. This is the second type of bond, right? What is the third type of bond? The third type of bond that we have seen here, this is between the nitrogen base pairs. So between the nitrogen base pairs, these are the hydrogen bonds, right? In this small segment of the DNA, you see three types of bonds: N-glycosidic bond between the sugar and the nitrogen base, phosphodiester bond between the phosphate and the alternating sugars, and hydrogen bonds between the nitrogen base pairs of the two respective strands. Okay, so these are the three types of bonds. Now let us look at the other features that we have already seen, so we'll elaborate them. Okay, all right, so what do we see next? Now let us see the other parts of the strand. So if you stretch it up and down, you can see So in the segment that we have drawn here, the strands have been unraveled, but in the other parts there is still the double helix thing that we can see here. So from this region to this region, you see there is a complete turn of one particular strand. So this one complete turn is called pitch of the DNA. This is equal to 34 angstrom. Okay, this length of this pitch. Now in this we've got 10 base pairs. Okay, 1 2 3 4 5 6 7 8 9 10 base pairs. So what will be the distance between each base pair? Distance between the steps or base pairs is 34 by 10 is 3.4 angstrom. Okay, so if you see this between this base pair here, that is the hydrogen bonds joining the nitrogen bases of these two strands, and this one here, what is the distance? This distance is equal to 3.4 angstrom. Okay, and likewise, so in this whole region there are 10 base pairs. Okay, also the distance between these two strands, this is 20 angstrom. That is the length of the base pairs. Okay, all right. So with this with this knowledge, let us now go back again. This topic in this DNA structure, it is Erwin-Chargaff's rules. Erwin-Chargaff in 1950 gave the Chargaff's rules. What did he say? So as per Chargaff's rules, the amount of purines is always equal to the amount of pyrimidines in a given DNA segment. Okay, so A plus G, adenine and guanine, is equal to the amount of thymine and cytosine. Okay, the second rule, it states that the amount of adenine